Richard is going to go first. So Richard, thanks for joining us. Richard is
You're
a
welcome.
postdoc at the moment at the Australian Institute of Marine Science in Townsville, that is in Queensland, in Australia. He just told us it's winter there and it's 25 degrees. So I'm jealous already. It's summer here. It's 17 here in London. Richard has worked mostly on reptile venoms, but on various other venoms as well. He's if you're not familiar with his work, check out this really cool series of papers on how neurotoxins in reptile venoms interact with nicotinic acetylcholine receptors. That's really cool work. But I happen to know that one of his greatest loves in the world of venom are the scaly, slimy ones, the venomous fishes. He's just published a comprehensive review paper on venomous fish, so go and check that out. From the top of my head, I don't know what is published, but I'm sure he mentions it in his talk. And it's venomous fishes that Richard is going to talk to us about now. So thank you for joining us. The digital podium is yours when you're ready.
Thank you. I just share my screen. Can you see that?
I can see that.
Yes. 1 second. Let me just, if this will move out the way. Here we go. Okay. So that should be better now.
Yep, perfect.
Right. So hi, everyone. I'm actually excited to do this talk because over here in Australia, we never actually get to listen to any of these talks because it's way too late by the time they're actually on. So yeah, so thanks for the introduction, Ronald. And as he made clear, I'm pretty fascinated by venomous fishes. And a lot of my work has actually involved reptiles, but my love is actually venomous fish. And so today I'm probably just going to give a kind of overview of the diversity of venomous fishes and essentially kind of run along the lines of the review that Ronald actually mentioned. So as most of you are probably aware, fish are vertebrates. And so if we just kind of put every single species of vertebrate into this small square here, right, around 55% of all species of vertebrates are actually fishes. Now, this is an insane amount of diversity when you consider that there's more species of fish than birds, reptiles, mammals, and amphibians combined, right? And so with this large diversity, we have a plethora of completely different adaptations that might arise. Adaptations, for example, such as different forms of locomotion. We have different body forms, different body plans. We have different dentition for different ecological niches, completely different sizes from very large to microscopic, different coloration patterns for different ecological purposes, defense, reproduction. And then, of course, what I'm actually going to talk about today, which is the adaptation of venom. So let's have a quick dive into the diversity of venomous fish. So maybe 20, 30 years ago, probably a bit more too, estimates of venomous fish, probably around 200, 250 different species. But now with recent work from Leo Smith and people like that, probably estimates of around about 3,000 venomous fish species. And these encompass probably around about 58 different families. And what's more kind of fascinating to me is that if we look at all these different venom systems in fishes, they've probably convergently evolved on at least 19 separate occasions. We have, at least 3 different independent evolutions within chondrichthyes, so your stingrays, sharks. We have a multitude of different independent evolutions within catfishes, and then kind of acanthioriforms, trichiniforms of weaver fishes. And then we have single evolutions in other groups. And the one most people are probably most familiar with when we talk about venomous fish are the scorpioniforms, so your stonefish, lionfish, and scorpionfish, for example. So how does this large diversity and evolving the venom system actually affect the diversification of these organisms? Well, this is actually work that I'd done a couple of years ago with the amazing Kevin Arbuckle, and we looked at venom across many different fish families, and we wanted to compare how diversification rates kind of change with the evolution of venom versus non-venomous families. And what we actually found was there was about a 2 times greater increase in diversification rates with the evolution of venom versus a non-venomous fish family. And our work was actually corroborated with previous work that me and Kevin had also done, where we looked at other venomous lineages, such as venomous snakes, and we also found the very same pattern. So diversification rates were increased within families as you evolve the venom system. And then there's work from another group, this paper over here called from Liu et al. And they found very similar kind of trend in increased diversification rates with the evolution of the venomous fang system in blennies. And so why might this be with the evolution of venom? Well, when you evolve a venom system, it can free from ecological constraints, particularly with things like defensive venoms, where you evolve a defensive venom, and so you decrease the amount of predators that are actually going to, target you, and so you are now free to kind of move into these new ecological areas, new environments, and you're more free to adapt to, new evolutionary traits. And so with this very large diversity, I'm sure, you can imagine there's a very, huge plethora of venom arsenal within fishes. So the most common one is what you're probably most familiar with, which is the venomous dorsal spines of ray fin fishes. And so the image on the right is probably the most common, which is essentially they have holocrine cells which are laden with venom. And so when these cells rupture, the venom is actually released into the wound where what the spine is caused. And then the one on the left is probably the one that everyone knows of, which is the stonefish. And these have a very derived gland system, more familiar to what you would see in venomous snakes, as an example. But this is actually a very rare evolutionary occurrence. And then we have other systems, kind of the stingray, which is they have a retroserrated detachable barb. And so this barb is actually designed so it's easy to penetrate into an organism. But then these retroserrations actually are very difficult to pull the barb out. And so you have more mechanical damage, which allows the venom to actually increase greater into the wound. And then you have the fangs of monognathous eels. You have the lower jaw fangs of Mecanthus blennies, and then you have this crazy alien-like teeth and buccal venom gland system of lampreys. We have clethral spines of stargazers. We have opercular spines of toadfishes and weaverfishes, and then we have the serrated pectoral spines of catfishes. And so you can see that with all these different evolutions, independent evolutions across fishes, we have this multitude of completely differently evolved venom systems, and it's fascinating to me. So how did venom in fishes evolve? Well, the short answer is we actually don't really know. But we have very good hypotheses, and also we can borrow kind of ideas from other venomous organisms. But what's most importantly is that defensive structures are already in place. So ray fin fishes, for example, they already have dorsal spines. And so all we need is kind of the recruitment of some sort of toxin or toxin system in place here. And so our most parsimonious explanation and the hypothesis that was proposed in around about the 1970s by, I think it was Anne Cameron and Robert Endine. And they proposed that venom toxins of fishes evolved from what we call ichthyocrinotoxins. And these are toxins that are actually present on the surface mucus layer of a lot of fishes. And they're usually used as noxious repellents or antimicrobial, antifouling compounds. And so the proposed hypothesis is essentially that we know that gene duplication events occurred a lot across fishes. And so it's possible that these ichthyocrinotoxin genes have undergone some gene duplication events. And then through processes such as neofunctionalization, we can have this freeing up of a gene copy, and it can now be utilized or recruited as a completely different function. And so ultimately, this is the best hypothesis we have, but rarely we don't know. And then even more importantly is that this only rarely applies to ray fin fishes. We actually have pretty much no idea or very few hypotheses of how other fish venom systems evolve, such as the stingray barbs or fangs and teeth in a lot of other venomous fishes. And actually, I threw this slide in actually at the last minute because I just wanted to give a shout out to this Ordovician conodon, which is potentially the very first venomous animal, and it is a very early fish ancestor. And so there's research actually back in 2009 which found that fossilized teeth of these conodonts actually have this groove in them, which is very reminiscent of what we see in venomous colubrid snakes. And so this groove actually is usually representative of venom being allowed to be pushed towards the tip. And so it would be really interesting if actually one of the very first venomous animals was in fact a fish. So what are the ecological functions of fish venoms? Well, as probably most of you are aware, because this is a venom-dominated research space, we could argue that there's three main functions of venom, ecological functions of venom, so defense, predation, competition. Now, obviously, it's not as black and white as this. There's a lot of gray area. There's a lot of nuance here, and, a lot of them can be multifunctional in a lot of respects. But for the purposes of this talk, we'll just stick with the main three. So if we kind of look across this tree of venomous groups, what we tend to find is the majority of groups actually only rarely evolve one function. So if you look across it, you have, most of them have only evolved for predatory or defensive purposes. There are very few groups that have actually evolved in multiple different purposes. And so if we look at bony fish here, for example, you can see that all three ecological functions have seemed to have risen. And again, that's testament to this huge diversity of venom apparatus within fishes. So let's go through each one. And so let's first look at defense. As I'm sure most of you are aware, the majority of fish do utilize their venom for defensive purposes. And it is mainly to cause pain. And I'm pretty sure most of you are kind of aware of how painful some of these fish venoms can be. So here's some testimonial accounts. And this first one is this guy over here on the right. And he says, I describe the pain as excruciating because the word comes from crucify, and that's exactly what it was. And then this second one is my personal favorite. The venom won't kill you, but it will make you wish you were dead. So I think they kind of tell you really how bad fish venoms actually can be in terms of pain. And so what's the point of causing pain as a defense? Well, in the short term, instant pain allows you to increase predator escape potential. So the predator is likely to let go, you can escape, live another day, reproduce, pass your genes on. But then there's also long term. So predator learned avoidance. So over time, predators become aware of how painful some of the stings are, and then they kind of utilize other characteristics such as warning coloration to kind of learn these predators and how painful their stings can be, and they just tend to leave them alone after a long period of time. So what causes the pain in defensive fish venoms? Well, the majority of pain actually comes from cytolytic or pore-forming toxins. So these break down cell surface membranes, and then you get a multitude of different bodily systems kind of firing off. You get kind of ions coming out of the cells and binding to receptors, and this is how pain is pretty much caused. And then you have pro-inflammatory toxins, where basically the pain actually comes through the inflammation system. And then we have pain mediators, so toxins that specifically target pain ion channels. But what about non-painful forms of defense? Well, some evidence actually suggests that there's some toxins within some fish venoms that actually bind to nicotinic acetylcholine receptors, and more specifically the alpha-1. And the alpha-1 receptor is actually more notable for at the neuromuscular junction. It actually allows for the contraction of muscles. And so I actually done research a couple of years ago where I looked at the binding of crude venom of stonefish. And what I actually found was the venom actually bound to alpha-1 nicotinic receptors. And now I guess a few of the pharmacologically more savvy people in the audience are probably like, well, that's not pain, right? That doesn't cause pain. So how is this a form of defense? Well, we actually hypothesized that the majority of predators of venomous fishes are sharks and rays, and these have very tactile mouths. And so they actually utilize prey handling with just their mouths alone. And so if you can paralyze this highly muscular and tactile area, it's more likely that they're going to let you go, and then you can swim away, you can survive another day. And actually, so it's possible that another form of defense is actually in causing paralysis of these sharks' mouths. And so this is an area that I'm pretty interested in, because I'm really interested in the ecology of venoms too. And so I'm hoping to kind of see where this research leads in the future. So moving on, what about predation? Mostly associated with lamprey, possibly monognathous eels and conodonts too, but we just don't know enough about the ecology of these fishes to actually make an assumption about it. So we'll go with lamprey for right now. They actually have a very parasitic lifestyle. So they attach to their host fishes and they blood feed for an extended period of their life cycle. So their venoms are actually pretty much dominated by anticoagulant toxins and chitinases. Chitinases are used kind of to break down that chitin within fish scales and just keep that blood flowing. What about competition? Well, this is actually probably one of my favorite examples of a venom system that's actually really cool in the way it's utilized and potentially the way it's evolved. And so this is actually from a venom from the Mercanthus grammistes, which is the striped fine blenny. And it was work done by Nick Casewell and others, like 2017, I think. And so what they found was the venom actually had these enkephalin peptides. So these peptides are actually, they bind to opioid receptors and cause analgesia and hypertension. But what they actually hypothesized was that the victims of the bites would actually cause disorientation and render coordination and swimming very difficult for the fish. And so what's really interesting about these fang blennies is they have very high competition for territory with other species of blenny fishes. And so what they hypothesized was that if the venom actually kind of got the victim high, they would be very difficult to swim, and so local predators could actually be easier to pick them out and take them away from the local environment, which would ultimately reduce the competition for the fine blenny. And so, if this hypothesis holds true, I think it's a very, very cool and interesting competitive venom system. So what about the venom composition of fishes? Well, if you ask some of the old school kind of toxinologists and the ones that haven't kept up with the research, they just kind of brush fish venom off. Oh, it's all cytolysins. And this actually might be true. And a lot of the early work on stonefishes, this actually holds true. But if we actually look at the literature now and we look and we kind of view venomous fishes as not just this scorpion forms and stonefish kind of research, then we actually find that the venoms of fishes are actually very diverse. So, of course, we have the cytolysins, which are the most probably highly researched, but we also find PLA2s, hyaluronidases, chitinases, lectins, kinins, yeah, cysteine-rich peptides. Then we have the enkephalins, small bioactive peptides, which are currently being, looked at for pharmaceutical purposes. And then if you look at some of the very old literature, we have uncharacterized toxins, which nobody's picked up this research since then. And we have monoamines and small molecules. And this is just a tiny amount of what we find in fish venoms. And so, again, going back to that rich diversity of venom systems within fishes, this is what you would expect. You would expect completely different, venom compositions across this, kind of these fish families. And again, we can't actually talk about venoms without kind of going into the bioprospecting, the bioinnovation kind of sphere. But as always, and I'm sure most of you are aware, it's always the needle in the haystack. There are millions of different venom and toxin compounds out there, but very few of them actually can, be repurposed for bioinnovation. And so, is there promise with fish venoms? Well, I believe there could be. We just don't really know. There's antimicrobial, antifouling compounds that are being investigated currently. And from what we know of the evolution of these toxins, this probably could be a very good sphere to look into. There's immunological disorders such as multiple sclerosis that is kind of, there's some peptides that are being looked at there. And then because of a lot of them are cytolytic, can we utilize them for anti-cancer as anti-cancer drugs and things like this? So we currently don't really know, and I think we won't really know until we actually see the fundamental basic research. And so on that note, I thought I'd put in a very cliche quote, which I actually kind of secretly love, and it's innovation is the key to the future, but basic research is the key to future innovation. And although it is cliche, it is so true to what we do as scientists. A lot of the funding bodies, they want the innovation right there, but without the very fundamental basic research, we actually don't know if the innovation can be there or what path to take to find this innovation. And so I just kind of, I guess, want to round out this talk by giving you reasons why I kind of love what I do in terms of researching venomous fish and the work that I'm actually currently undertaking. So fish are cool. So why wouldn't I or anyone else out there want to research them? As I hopefully have shown through this talk, the diversity of fishes is insane. And there's so many different adaptations, not just within venomous fish, but in fish as a whole. And so there's so much we can actually learn about them. We know very little about the venom evolution and the biochemical makeup of their arsenal. And I think by understanding fish venom evolution, that can also, help us to understand other venomous fish groups, such as how, snake venom evolves, for example, and scorpion and spider venoms, and even other marine systems that we just don't have access to. Fish can teach us a lot about how complex traits are evolved. One fascinating area to me in terms of when we study venom is that as anti-predator defense evolves, we tend to have this conjunction of other traits evolved in conjunction with it. So things like aposematism, mimicry, we have, bright colorations and camouflage. And so it's fascinating to me to actually be able to understand how all these complex traits actually come together that surround just this one anti-predator defense. And I think fish are actually a very good model that haven't been utilized in this research space. And we actually need better reasons for conservation. And again, I know this is kind of cliche, which I'll probably talk a bit more about in the next few slides, but we actually need better reasons for conservation. And if the potential of the next wonder drug is that we need to conserve these species for that potential, then so be it. So what are my current research focus and interests? Well, currently I'm in the venom sphere anyway. I'm currently focused on Australian stingrays. We actually know very little about their venom. The majority of work on stingrays has actually been in freshwater South American species, and the research looks promising there. How this transcends to marine Australian stingrays is still unknown, but I'm working on that. And stingrays have a convergently evolved system very different to that of the majority of research fishes, which is the ray fin fishes. And as I showed earlier, they have a completely different venom arsenal. So they have that retroserrated barb that actually detaches from the body and causes a lot of mechanical damage. And so again, looking at the evolution of these stingrays and their venom system is of high interest to me. Is there bioprospecting of the venom? Well, as mentioned, the freshwater South American species, there have been quite a few small peptides been actually found in the venoms. And so there's potential there for bioprospecting if that translates to Australian marine stingray species too. And can we help in conservation efforts through venom research? So Australian stingrays are currently, there's a few species that are actually endangered and potentially close to extinction. And so if we can utilize kind of our edge on the venom research and say, hey, there could be the next wonder drug come from these venoms, but we actually don't really, without conserving them, we can't figure out whether this is the case. Everyone loves a good, cute animal, and stingrays are not cute. At least the majority of people don't think so. And so if we can utilize conservation through another avenue, then this would be great, not just for our own efforts, but I guess for the reef systems and the marine sphere in general. And so I'd like to just end the talk by saying thank you for everyone who came along today. And if anyone's interested in what I do, there are some of my contact details, and I'm happy to talk. Thank you.
Thank you, Richard. That was a great talk. You have beautiful slides. I'm thinking, because I'm a fan of the white background and the black font, but I'm now thinking maybe I should change. So
I thought I'd stick it with blue for the marine.
I really like it. People, if you have questions or comments, this is your chance. You can type them in the chat, or you can turn your camera on and speak.
Hi, can you hear me?
Yep.
Hi.
So I was wondering, you were mentioning how some of the venomous fishes, they store the venom in the cells in the venom gland before it's in the dorsal spines. Yep. I was just wondering, how are these like toxins stored? Are they in some kind of vesicles, or are they in an inactive form? Has there been any research into this?
Yeah, so it's very old research actually, and it depends on what system you're talking about, because there's the two dorsal systems. There's the the most prevalent one, which is essentially it's just a bunch of holocrine cells. And I'm not sure if you're aware what holocrine cells are, but holocrine cells are basically, they have to rupture to release their contents. And so the venom's actually stored within these holocrine cells. And what happens is when the spine penetrates into an organism, these cells rupture, and that's how the venom is kind of released. But then there's the second dorsal spine system, which is most common in stonefishes, which is the actual gland. And so the venom's actually stored within the gland, and the cells within the gland actually release the venom into the gland to be stored, and then the venom actually goes up through a duct once it's depressed, and that's how the venom goes into the system. So there's very old work on histology of these systems. And one thing that I actually highlighted in my review is that if you look at the literature, these two systems are very different, but the way people talk about them, particularly in the past, and the way they've referenced them, it's always just referred to as venom gland. And that's not, I don't think that holds true. I think we have one strict venom gland and then another system, which is holocrine cells, which is completely different.
I've got a question too, if other people don't have one. Are you aware of any fish venoms being at the center of antagonistic co-evolution, like some predatory venoms are, where you have, as a prey animal, say you're a ground squirrel in California, it's incumbent upon you to evolve resistance to the most immobilizing components of rattlesnake venom, because otherwise you're effed. So is there anything known about predators of venomous fishes being in a similar sort of arms race?
So no, the short answer is no. We actually don't really know much. However, because the way defensive venoms evolve is that usually it's very difficult to actually kind of evolve resistance, at least biochemical resistance anyway. You can get defense kind of, I guess, physiological resistance in terms of, like, thicker skin or whatever. But for defensive venoms tend to usually evolve to have a wide range of kind of targets. And so, like, for example, pore forming toxins, how do you evolve something that actually stops pore formation, right? It's literally just breaking down the cellular components of the cell. So in terms of defensive venoms, I'd say, or defensive venoms that cause pain, I'd say it's very unlikely. I'm not saying impossible, because evolution does kind of strange things. In terms of predatory venoms, probably not. Again, and again, there's just not really much data. Like the ecology of a lot of these fish species, we virtually know nothing about. Like I gave the example of, we assume that the fangs of monognathous eels are for predator purposes, but we actually really don't even know their ecology.
I mean, that was that picture you showed. I don't know where you got it from, but that's a beautiful picture, because mostly the images I've seen in literature are these, when they describe a new species of monognathous eel, it's just a line drawing with a pointy bit. You go, I wish they showed more. Yeah, no, it is specifically the defensive use. I was also thinking, defensive venoms are such a different beast compared to any other kind of venom. Any other kind of venom, whether it's for blood feeding or predation or competition, is proactively used as a tool to initiate interactions with other organisms. Defensive venoms are the complete opposite. So they basically have one purpose: leave me alone.
Yeah, pretty much.
They're not going to bring it to a target. And the response is different too, because if you bite into a painful fish, you're going to go, I'm going to just eat something else. Whereas if you are the ground squirrel, the snake will keep coming back at you. So I think the whole system of rules according to which these defensive venoms evolve, not just in fish, but in other defensive venoms as well, is completely different compared to proactively used venoms. Anyway, that's just I'm thinking about that. So I'm not that surprised that
I think so. So resistance can evolve to pain venoms that target, specific ion channel receptors. So there is evidence, not in fish, but in other venomous organisms. But I think in terms of generalized kind of pain, where it's kind of, targeting the immune system or targeting cells, I think it's very difficult to get resistance from.
Yep. Harriet, would you like to ask a question? I'm going to make you the last, and then I see that there's also a question in the chat, but maybe Richard can answer that as Andy gives his talk.
Yes. Hello. Can you hear me?
Oh,
Okay.
yeah. Oh, sorry,
Can
Harriet.
sorry. Can I ask?
Sure.
Yes. So I have two questions. First is about the paper about the varicose venom, because you mentioned there that you also tested a CV1.2 L-type voltage-gated calcium channel, but only in a screening assay. And I wanted to ask if you further continued with this toxin on patch clamp or other more high resolution.
No, so short answer is no. So the system I used for are you talking about the paper with the nicotinic receptors?
Yes, and also the CV1.2.
Yeah. So essentially we use mimetopes. So it's kind of a synthetic peptide that actually represents the orthosteric binding site of the nicotinic receptor. We didn't use whole receptors because you wouldn't be able to use kind of the whole crude venom with that, because it would just cause pore formation and just destroy the cells anyway. So it would be kind of pointless. You would have to kind of isolate the toxins. In short answer, I would actually really like to go further with that research in the future, but currently that paper was some time ago, and yeah, it was just because I had the assay at hand and we just tested the venom to see what was potential, because there was early work to show that there was potentially nicotinic receptor binding targets.
And if I may have one more question, do you know if the alpha-1 subunit of the nicotinic acetylcholine receptors is different in comparison to humans, or is it evolutionary conserved?
So across multiple different species, you do get a lot of amino acid changes within the orthosteric site, and particularly the orthosteric site is where you're going to get the majority of action, and likely actually a lot of my work showed that's where a lot of resistance elements evolved. And so amino acid changes across multiple different species and different families is very different. And so the ones I tested were for shark, fish, and I can't remember the other species, but yeah, so they are very different than humans. And so again, you're correct in that it could actually not even affect human receptors, but might be more specific to predators such as sharks.
All right, thank you very much.